Until recently, family historians had to rely on written documents and word of mouth to piece together the story of a family. And science has now provided us another tool, and I've been amazed at what we've learned from the Yoder DNA testing over the past 10 years. This presentation is intended to give you a bit of an overview. Have any in this room been a part of the Yoder newsletter DNA test? Got several? Yep. Appreciate that, your support, and we'll kind of summarize what is shown. I'll begin by trying to explain how DNA testing works and how it's interpreted. I'll share some of the results from the Yoder test, including a standard profile, which goes back to our Steffensburg ancestors, a separate Amish mutation to this profile, which appears, what the 100 marker profile looks like, and I'll have to give you some terms before you understand that one, for our most recent common ancestor. And that person was a Carl Yoder born in 1548. And I'll also talk about some of the surprises that greeted us through the testing. Now, DNA testing is of several different types. One of them is autosomal, that looks at your whole DNA makeup and tries to figure cousins or relationships on your father or your mother's side. It doesn't really matter. Then there's mitochondrial DNA testing that basically tracks back a female line and somehow tells you if you got a common female ancestor of some kinds. I don't really understand that. What we did is testing of the Y chromosome, which is the sex determinant chromosome, and it determines whether a child's going to be a boy or a girl. And the boy's going to have an X and a Y chromosome. A girl's going to have an X and an X. So it's the man that determines by giving one or the other what the gender of the child is going to be, because the woman has an X and an X. The Y changes very little over hundreds of years, and there are markers or positions on that chromosome which can be measured and that give it a unique fingerprint. People with matching DNA fingerprints may have a common male ancestor, almost certainly if they share the same surname. We partnered with a firm called Family Tree DNA, which is the company that worked with National Geographic on their Human Genome Project. And I'll give you some vocabulary words now. First, DNA is deoxyribonucleic acid, and that's the basic structure of heredity. It's the material in our cells that contains our genetic makeup. Genomes, let's go down a bit. Genes are the basic units of inheritance, are found on strings called chromosomes. The humans have 23 pairs of chromosomes, and as I said, one of them is the sex determinant, XX and XY for men and XX for women. Specific physical locations on the chromosome are called markers, and these can be measured and given a numeric value. A haplogroup, first, a combination of the marker values at selected locations of that chromosome give what's called a profile for the Y DNA. The haplogroup is a more generic grouping based on what's found in the testing that links you to an ancient family of humanity, a tribal type linkage that goes back maybe many thousand years. Now, here to summarize again, the Y DNA comes through the male line, grandfather, great-grandfather, grandfather, so forth, down to brother. And that is what we tested because we're looking particularly at the surname, and we want to see what it tells us. When they lay out and measure and compare people's Y chromosome DNA, they look at these specific locations and they compare for a match. And it's like a string of beads. If this example, A matches B at number 4, C at number 4 doesn't match. The value or the measurement number associated with that position in the marker is called an allele. Now, when we do a testing of the DNA of a person living today, the initial testing we did was like 12 markers, and we've increased that over. But let's say each of these blocks is a different individual known to have a common ancestor. Well, you see 14, 23, 16, 10, 15, 16, 11. The difference between him and the one in the middle is 16 instead of 15. Well, that you know that because they got their DNA through a string that started with the same guy, that was the source of the original values that came down in the family. So all the markers that match between these people, you can say, well, that common male ancestor had that value in his marker, even though he may have been hundreds of years ago. When you have one situation where you're comparing two descendants through separate sons and they have a difference, if you can find a third son and look at his values in the measurements, that's the tiebreaker. The odd man out is the line in which there was a mutation, and you can say that the common male ancestor's DNA profile for the Y chromosome at these marker numbers, and that DYS whatever 393 is the name of that location on the chromosome, value 14, that's the common ancestor's profile. So people ask, well, how can you tell someone 300 years ago what his DNA was? You do it by this process of different sons, testing today, and then comparing and eliminating where the odd man out is, if you can. Now, if you've got 3 situations where they're all different, then that particular value you may not be able to say this is what the common ancestor had. But you, in large part, you can, even at 100 marker comparison. What the kit is a little toothbrush-like thing. It comes in a mailer. You scrape off some cells from the inside of your mouth. You put it back in the mailing package. You break off the head, bottle it up, put it in the mailer, and you send it back into the lab. University of Arizona is the one that does the testing for National Geographic and for Family Tree DNA. We've completed over 100 tests under the sponsorship of the Yoder newsletter. We paid for quite a few of them out of our kitty. North Carolina Yoders and the Ole Yoders, they funded some of them. A lot of individuals have paid for their own tests. Again, the haplogroup that we fall in is one called I2A2A-M223, and that's the scientific name for the group. They've changed the naming process over the last 10 years a couple of times. It occurs in moderate frequency among the populations of Northwest Europe, and it peaks in Lower Saxony and Central Germany with offshoots in Romania, Moldavia, and Russia. And it is not a majority in any part of that area. It's a lesser occurrence. We tested initially at 12, and then we upped it to 67 markers, and this is the common male profile for the common ancestors from both the Steffensburg, the Reformed Church, and the Amish Yoders, and what their 67 marker DNA looks like. If you go to the website for the Yoder newsletter, there's a a button partway down. It's not kept completely current, but you'll see a spreadsheet with even more comparisons by family groups. Our first major discovery was this fact that there is a difference in the Amish profile, and that goes for the Yoders of 1742 and the Amish coming in the 1800s, and that is that at the third position measured, what is called DYS19, instead of a 15, it's a 16. And that becomes the fingerprint that helps distinguish things for us between the two branches. Again, this is the tree, the basic tree that shows the relationships. Hans and Yost, they're probable cousins, see the question mark. Hans of Great Swamp, the Mennonite. Their brother's son Conrad, Conrad's nephew Melchior, Michael, whose father Samuel wrote to Schweitzer Christian in Somerset County and called him Mein lieber Vetter. Joseph Yoder of Bureau County. These are descendants who have been tested. They have Amish profiles, meaning the one character difference. All of these fellows have one character differences. We know enough about the family of this Casper in Germany to see that he's got a son named Christian and a grandson named Christian, and the family's pretty well fleshed out. The same Christian couldn't have been on the boat in 1742 as the one that was a bishop in Germany 10 years later. So we know he is ruled out as being the father of our Amish line. But any one of the other males in the family of Yost could have been, and they need to be, to point back that DNA to Yost. Now, this is a comparison that I thought was kind of interesting. At 67 markers, you may recognize this fellow right here. His DNA profile at 67 markers is one measurement different, like it's an 11 instead of a 12 at one of those 67 from the late Hubert Yoder in North Carolina. It's two difference from Joe Yoder of the Ole Valley Yoders. It's three marker difference from a member of the Melchior family. And it's three marker difference from a member of the Hans of Great Swamp family. This is Ken Yoder's late brother. This is what the 100 marker profile looks like. The top line is the most recent common ancestor, MRCA. Well, we know because so many different directions that 16 versus the 15, that 15 is the value of the next generation back, or Nicholas couldn't have had it, and Andreas couldn't have had it today. It changed with Yost. Don't expect you to digest any of this. You'll see some places where there's a variance in one line or another between the Amish, Conrad Yoder, Hans of Great Swamp. Out of 100 markers, there's supposedly about a 2% chance of a variation in any one generation. But you saw how infrequently it occurred. We did get some surprises through the testing. And this fell in the generation of some grandsons of Hans Yoder of the Ole Valley. His son, John, had 4 known sons. One of them matched the Steffensburg profile. One of them had girls, and we can't test them. 2 others both had different profiles from the Steffensburg, meaning also from the John, the father, and also from each other. So there were either some adoptions or some other unknown event that took place. They were believed to be, and in the wills are counted as the children of OH1 John, but they didn't have the same father, and their father wasn't John. Now, that's kind of been a quandary for the people that discovered that, but it has been very useful in identifying some of the missing links lines of Yoder's down into the 1800s, and we've done that in several cases. Because of the DNA, we've been able to trace back to one or the other, OH13 Samuel or OH14 Peter. We also had some surprises. There's a family named Yoder and Yater. We tested them just to check out. They match to each other, but not to the Steffensburg Yoders, although I did say that there are some families in which the Yoder has gone to Yoder, and they do confirm with their Yoder DNA profile. Here's different groups of Yaders. They all apparently support the belief among that family that these were the sons of a Johannes Yoder in Stuttgart, Germany, who came to America. Another surprise is that there was an Andrew Yoder/Yoder, depending on the years in which his family, in fact, even a Yoder, Y-U-T-E-R, what years the census was taken. This fellow, weren't sure whether it was a Yoder or what. He ended up being a Yoder based on the DNA. It matches to the Steffensburg profile. We don't know exactly how he fits in, although there was an Andreas Yoder in the Moosbach area in at least one record about the time that he was reportedly in the United States. Another surprise, there was a family of a Yost Yoder, Y-O-T-T-E-R, that married in 1758 in Lebanon County, Pennsylvania. He had some children that went in different directions, one of whom went to Sussex County, New Jersey. And we have a test from a gentleman who's a Yoder, whose family does seem to link to that family, and he comes up as a match to the Amish profile. So that's a surprise for us and something that hopefully someday we'll understand. Another surprise is the name Yadi, is fairly close in the DNA profile to the Yoder family. And they're the first immigrants, Peter and Ulrich Yordi of Lancaster County. They came around 1717. And a current descendant of Peter Yordi of 1770 matches at the 25 marker compare 23 out of 25 markers, which according to the information on the test site says there's a 99% likelihood of a common ancestor. So it may well be that the first Yoder to run for president of the United States was Mayor Sam Yordi of Los Angeles, who ran in 1972. A member of this family has speculated that given the amount of differences in a further test, it may be several hundred years further back where there's a common ancestor, but that is a possibility. We also found that a Zimmerman, whose ancestries came out of Steffensburg, was close to the Yoders. And remember I said that surnames were not used in Europe until around the year 1200. So there are male ancestors without surnames in generations and back, and so this Zimmerman and us Yoders may very well have a common ancestor at some point. Here's a summary of the charts of the differences. Just about everybody's a 15 except for us Amish guys like me, us ex-Amish descendant guys like me. And our conclusion is that it's been a very successful tool for us in augmenting what we know in books and facts and church records and so forth. And while it may not necessarily answer all the questions for us, it helped us answer some.